Right, good day junior tickies, and welcome to this video where we're looking at worksheet question 4 in our preparation for our exams. My name is Mr. P. We're going to be working through this question together and hopefully giving you a better understanding of the work and how to approach exam style questions. So we're looking at question 4. And the first thing we need to do is we need to get context. So I tell my students this all the time: before you look at the questions, look at the diagram that you are given first. In this case, it tells us the diagram below shows a cell undergoing a specific stage of meiosis. So we know the process here is meiosis. And if we look carefully, we can see that we've got replicated chromosomes, homologous chromosomes, particularly now being pulled what looks like to opposite poles of the cell. This is meiosis, so I can assume that. I've got two different sized chromosomes above and below, and this is going to obviously let me or lead me to the types of questions that are possible in this specific image. We see that there is a label A to my diagram as well, and in getting context of my image I can see that that's a spindle fiber. So it's most likely going to be one of the questions I'm going to be asked in question 4. But here, let's have a look at question 4.1. If we're going to have a look at 4.1, it says to us, state two characteristics of homologous chromosomes. So if we see the homologous chromosomes over here, particularly, I've already identified that there are genes that are identified on each of these homologous chromosomes, in each of the homologous pairs as well. So we can say that, or we can refer to these genes as being the specific size and shape, but also the fact that they carry the same genes. These homologous chromosomes carry the same genes at the same distance from the centromere. Okay? We refer to this distance from the centromere as the loci. So they have these alleles at the same loci on each of the chromosomes. The centromere position is also the same. If we look at identifying structure A, we did that when we got some context to this image, and we can see that we have spindle fibers here. If we're looking at question 4.3a particularly, it says how many of each of the following is present in the diagram. How many chromatids? Well, if we look at chromatids being joined by a centromere, I have 2 on each one of those replicated chromosomes. Therefore, I'll have 2, 2, 2, and 2. And if we add these all together, we have 8. 8 chromatids. Now, how many centromeres do we count? Well, we can see that there they are. I've colored them in purple, and we have 4 centromeres. Now, question 4.4 is becoming more common in the exams that we see. It says to us here, draw a labeled diagram of one of the cells that would result at the end of telophase 1. So now we're looking at the conclusion of meiosis 1 and not meiosis 2. So if we're looking at telophase 1 and we're drawing only one cell, we should be able to do this quite easily. There I've circled the 1/2 of the meiosis taking place here, because in telophase 1, that is what I'm going to need to illustrate in one of the cells that would result after telophase 1. And if I draw it, I would draw it like this, because at the end of telophase 1, I'm going to have the reforming of the nucleus. I'm going to have my full chromosomes that are or that have 2 chromatids still joined with a centromere, and this is going to be what I see at the end of telophase 1. Now it says to us draw a labeled diagram. So there I've drawn my diagram, and now now we have to label this. It's my responsibility not to forget the other part of this question, where we now have to label. And there I can label my chromatids as part of a chromosome. I can label my nuclear membrane that is reappearing, the cytoplasm, the centromere, and the cell membrane. Right, junior tickies, I hope that this has given us a bit of insight in how to approach these questions. I see that I have labeled the cytoplasm twice there, but rather label it twice than forget to label it at all. So moving on from here, we're going to be doing question 5 in a separate video. Please join me for that. I do hope that you've enjoyed this video and it's helped you to a certain extent in how to approach questions like this. Make sure you get your hands on the worksheet as well as the worksheet memorandum and the theory booklet, just to help you prepare as best as possible for this section on meiosis in your final exams. Thank you, Junior Techies.